Our study, Brain Decode Deep Nets, is to use human brain to explain how does deep network work. But first, how does human brain solve visual recognition? When we look at this image, we recognize it's a photo of two zebras. Our brain is multitasking. Inside the brain, it does edge detection, segmentation, classification, and language caption. Also, visual brain is organized into regions. Each region has specialized functions. For example, V1 does edge detection and orientation filtering. This image shows V1 feature is on edge detection and counters. FFA is face selective. If there's a face, neurons in FFA fires. This image shows FFA feature focus on human face and also the fish face. In the brain, image processing and feature computation are organized in a hierarchical fashion. V1 is the initial visual region that physically connected to the input from the eyes. Representation in the brain becomes increasingly abstract from V1 onward to other regions. It's somewhat like a feed-forward deep net. Next, we recall ways to study how does deep net compute feature over layers. One way to find out is to prop layer from pre-trained model to some tasks. For example, we can take a pre-trained model and run a grid search on which layer best predict an edge detection task. We find the early layer has the best score and conclude that early layer is doing edge detection. Then we could repeat the same process for another task, like segmentation, and find out middle layer is doing segmentation. This simple experiment tells us something about feature computation in deep nets. We need more tasks. Where do we find more tasks? It's the brain. Brain is massive multitasking. We can think of each brain voxel as a task. There's edge detection in V1, body detection in EBA, face detection in FFA. Brain is massive multitasking, and these tasks are organized from low level to high level in a hierarchical way. Now we have a pool of tasks from the brain. We do the same grid search and find out which layer best predict each brain voxel. Then we color the brain voxel by the index of deep network layer that predict this voxel. We can see from the left side image, brain region from low level to high level are mapped to early to late deepnet layers. This searching is the basic idea and motivation behind this work. But instead of doing research, we design the model such that each voxel automatically selects which layer to use. The brain data is captured by fMRI experiments. The participants look at a screen, and the screen shows cocoa images. For this study, we're only interested in the surface of the brain, not subcortical brain response. As the left side video shows, the brain surface is flattened, so we can plot the 3D brain into 2D. After processing the fMRI scan, each brain scan is matched with the image shown in the bottom right video. The goal of the brain encoding model is to predict brain response from the given image. For the model, given an image, we use a pre-trained vision model to extract features. The brain encoding model we are using is a simple linear regression on the features. The key is how we select feature for each voxel. The entire VIT feature space can be viewed from 4 axes. Layer is typical 12 to 40 layers. Space, each element is a spatial patch. Scale, because the special class token in VIT is global scale, other tokens are local scale. And channel. The key is to trim the model to pick the best feature for each voxel. Each voxel then a weight for every layer. Features are weighted sum over the layer axis. And each voxel then a coordinate on the space, and they do bilinear interpolation to get the feature. Each voxel also then a weight for space versus class token and sum them. Finally, on the selected channels, each voxel uses a linear regression to predict its brain response. This video shows the feature selection during training. Before this work, people predefine and handcraft the feature selection. In this work, the feature selection automatically emerges over training. Another key aspect of our method is that we use topological structure of the brain to improve the feature selection. The motivation is that spatially closed voxels have similar functionality, and therefore they should select similar features. In practice, topological constraint is implemented by an MLP that takes voxels coordinates as input and output the selection weights. This MLP enforces local continuity and forces voxels with close by coordinates to select similar features. Because data from a single brain voxel is highly noisy due to the nature of fMRI, but we can greatly boost the signal-to-noise ratio by considering neighborhood voxels. Without topology constraint, the brain-to-network mapping will be noisy and highly uncertain. In this image, black means a low confidence soft selection, which means a uniform average of all layers. Now we have the model. The model showed a mapping between brain and deep nets. Next, when looking into different deep nets, their brain mapping is different. The first question we ask is, how does network training objectives lead to different learned models? We have a variety of supervised and unsupervised training network models. All their last layers are shown to be effective for image depth prediction, but their intermediate layer features are not fully evaluated and remain mysterious. Our brain-to-network mapping gives us a way to look into the hidden layers. For a fair comparison, all the models are VIT architecture. First, we look at the three supervised models, CLEAP, ImageNet, and SAM. The CLEAP mapping showed a clear hierarchy. Early visual brain region mapped to early network layers in blue, and high-level brain region mapped to the late layers in red. The last layer is mapped to the brain region that performs the task the model is trained on. CLEAP last layer is mapped to higher level brain region. ImageNet last layer is mapped to the middle level brain. Also, SAM last layer is mapped to the middle level brain. The fact that ImageNet and SAM last layer map to the middle part of the brain suggests that there's bottom-up feature computation in early to middle layers, then top-down task prediction in late layers. ImageNet and SAM training objective is a middle-level task, but their middle layer compute high-level features. Next, we look into three self-supervised models, Dino, MAE, and MoCo. Dino is learned with self-distillation, contrastive learning, and mask reconstruction. However, Dino's last layer has not been selected in the brain prediction. This is also true for mask autoencoder's last layer. It does not align with any brain region. We could infer that mask reconstruction task is not a brain-related task. Momental contrastive learning MoCo last layer is aligned to the higher level region brain. MoCo's second last layer aligns with the ventral stream, but not the dorsal stream. Ventral stream is the what part of the brain, dorsal stream is the where part. This means contrastive learning is much more about semantics than spatial relationship. Next, we look into what happens if we scale up the model size but use the same training objective. We start with the base size 12-layer model. With the base size Dino model, we see that early brain is aligned to the middle layers. As we increase the model size to 24 and then 40 layers, we see less alignment for the early layers and more clutter on the late layers. This demonstrates that Dino larger model is actually wasting computational resource on the early layers. A similar observation can be made for the larger SAM model. The larger SAM model late layer misaligned from the brain, suggesting that the late layer are doing something unrelated to the brain. When model size scale up, all of the models we tested had worse brain alignment. The only exception is CLEAP. CLEAP had better brain alignment when scaled at model size. The best brain alignment of CLEAP is CLEAP extra large model. Next, we look into fine-tuning. How does fine-tuning change the layers? Our hypothesis is that the hierarchical organization of the brain is a template for adaptation and generalization, and a computer model that aligns with the brain should also be able to adapt to dynamic tasks better. We fine-tuned the models to two small datasets, medical imaging and satellite imaging. For Dino, after fine-tuning, its late layer does not align with the brain anymore. Dino changed the later layers. The late layers are redirected to the new task and completely forgot its original functionality. This demonstrates catastrophic forgetting. The same rerouting and forgetting happened to Sam. After fine-tuning, Sam's late layer lose alignment to the brain. This means fine-tuning completely reshaped Sam's late layer computation. However, CLEAP showed different result. CLEAP maintained its brain alignment after fine-tuning. CLEAP is able to adapt to this new task without forgetting existing computation modules. Recall that CLEAP also has the best brain alignment, especially when scaled up. Therefore, we conjecture that better brain alignment means better adaptation to dynamic tasks. Finally, we look into channel selection in brain mapping. Neural network channel mapping offers a more detailed picture of network to brain mapping than layer mapping. Channel mapping is obtained by clustering voxels that have similar linear regression weights. We make several empirical observations. Early brain regions tend to share a few similar neural network channels. Higher level brain regions use a more diverse set of channels. For Clip and Dino, their early brain channel is symmetrical for the left and right hemisphere, and also symmetrical for the ventral and dorsal visual stream. While the early visual brain had used similar channels, it used diverse spatial locations as shown by the space selection. However, for SAM and MAE, we find their early brain channel is not symmetrical. Asymmetry means SAM and MAE's channel are changing across space, and therefore not shift invariant. We can also use the known function of brain region to visualize the selected channels. For each brain region, we generalize across all voxels inside their channel selection weights into three principal vectors. The top three PC creates an RGB image of the channel response. From this feature map, we can see V1 selected edge detection features and FFA selected face features. We can also compare channel visualization across different models to see how well they captured semantic information. This image shows more examples from face selective brain region FFA. Features are on the face image region. Interestingly, dino face feature generalize across human and fish. This image shows more examples from body selective brain region EBA. Features are on body but not including the face. We can segment out the human body from clip and dino V2 feature. SAM and MAE has a poor body feature. In summary, brain scan is dense annotation to images. We use mapping between DeepNet and brain to look inside DeepNets.